Hello! In this video I'm going to give a brief introduction to histology. So histology is the study of tissues. On the screen here we actually see a tissue, and it's something that people don't often think of as a tissue in the human body. What we're looking at here in this picture is blood. Most of the cells that we see here are red blood cells, but right in the middle we have what we call a white blood cell, even though it's not white. And then over to the side here we have a couple of platelets. And this is a tissue. A tissue is defined as a group of cells working together to perform a particular function. And usually it's a group of similar cells. So blood is one of a few fluid tissues in the human body. So as I mentioned, the definition of a tissue is a group of similar cells working together to perform a particular function, whether that be to transport something, or connect something, or to protect something. Histo means tissue, so histogenesis is the production of tissues, and this is something that happens when we're an embryo. Histology, then, of course, is the study of tissues. So why do we have to use a microscope to study histology? Well, when we're studying histology, we're often looking at the cells that are part of the tissue, or we're looking at the stuff between the cells. And in order to do that, we can't use our naked eye, we have to use a microscope. Most of the time when we're looking at a tissue under the microscope, what we're actually looking at is a small piece of an organ. Right? An organ, by definition, is a group of tissues working together to perform a particular function. So when we look at an organ, we're going to see multiple tissue types. Therefore, if we're looking at a small little piece of an organ on a microscope slide, you wouldn't expect to find only one tissue type. You're going to expect to find multiple tissue types. A lot of times when we're studying histology, my students will say, "Carrie, can't you just show me a slide with just areolar or connective tissue on it?" And I say, no, I can't do that because areolar connective tissue exists as parts of organs. So I have to show you a slide of an organ, and that contains multiple tissue types, and we have to look for a particular tissue within that slide. In histology, the specimens are fixed or preserved, which basically means chemicals have been added so they don't rot. And then the organ has to be thinly sliced or sectioned so that light can pass through. If the particular piece of organ or tissue is too thick, then when you put it on the microscope slide, the light can't pass through and you won't be able to see anything. So it has to be sliced very, very, very thin. Not only that, but the specimens are also often stained, because without stain, you really can't see much of anything for some tissues. Looking here on the screen, we see what kind of looks like maybe water droplets or something. It's kind of hard to tell. What we're actually looking at are cells. Right? So here we're looking at the cells when they're stained. You can clearly see where one cell ends and another one begins, for the most part. You can see cytoplasm, you can see nucleus, you can see a lot more details. Whereas here, not so much. So most of the tissues that we look at are going to be stained so that we can better see the structures of the tissue. Interpreting tissue sections can be very difficult, and this is something that even professionals still struggle with. Because when we're looking at a microscope slide, what we're looking at is a two-dimensional picture that's coming from a three-dimensional object, and it's very difficult to take that two-dimensional picture and build it into the three-dimensional structure. To give you a few examples, in this first picture here we see this slightly oval-shaped white structure, right? What is that? Well, let's make cuts on the structure along these dashed lines, and then we see this. And now that we've seen all of these different sections from the structure, we can see, oh, that's a hard-boiled egg, right? But just looking at these structures here, would you necessarily have known that that was a hard-boiled egg? No. Now maybe the bottom structures, yeah, because you can see the yolk, you might have known that was a hard-boiled egg. But the upper structures, not so much. So it's kind of difficult to build this into the three-dimensional object. Now looking over at the macaroni noodle, very clearly we can tell that that's a macaroni noodle. But now let's make cuts along the dashed lines, and we're going to see this. Now, if I showed you the picture with the red background there, if I showed you just that in isolation, would you have gone, aha, that is definitely a macaroni noodle? No, you wouldn't have done that, right? Probably if I would have shown you any of those pictures, the green or the blue, you wouldn't have known it was a macaroni noodle. But now that we can see the three pictures together and we know that it's coming from the macaroni noodle, you can kind of build that together, that picture together in your head to develop that three-dimensionality. But again, it's difficult. This is a difficult thing to do. The last picture is a duct. That's D-U-C-T, not D-U-C-K. A duct is a small tube that we'd find in the human body. And we're going to find these in lots of places. For example, we have ducts that go from our sweat glands to the surface of the skin, and they're there to deliver the sweat to the surface of the skin. This is what a duct often looks like when it's been cut. And if you notice that it doesn't look like a continuous tube, does it? And that's because the tubes in our body are generally not flat. They have dimensionality. They move side to side, front to back, right? And so when you cut it, it's not going to look like an exact tube. And again, this goes back to the idea of taking this two-dimensional picture and building it into the three-dimensional object. What also makes interpreting tissue sections difficult is the fact that we're often looking at an organ, and organs by definition contain multiple tissue types. So you have to keep in mind that you're going to be looking at more than one type of tissue. So over here in this picture we see a micrograph, which is a picture of a microscope slide. And this is a micrograph taken from skin. And you can clearly see there are some different things going on in this picture, right? There's more than one tissue type here. Knowing the orientation that a cut was made of an organ can often be very helpful for understanding what you're looking at on a microscope slide. For instance, if you take a look at the bones over here, right? One bone was cut lengthwise, we call that a longitudinal section. One bone was cut horizontally, what we call a cross section, and then the last bone was cut at an angle that was not longitudinal and was not horizontal, and we call those oblique sections. And if you look at the cut bone in each of these, they look quite different. And so knowing the way that a cut was made can be helpful for interpreting a particular tissue. Right? That longitudinal bone looks very different than that cross-section bone. The same is true for tubes. When tubes are cut, it helps to know their orientation. So the other pictures are showing a series of blood vessels cut at different angles. One a longitudinal section where you just see two lines when it's cut. Then one in a cross-section where the blood vessel will look like a circle. And then we have the oblique section. And speaking of tubes, it's also important to know that a lot of times tubes collapse once they've been cut. And this is because generally it's the contents of a tube that hold the tube open inside the body. So once you take the tissue out of the body and start cutting it, the contents of the tube are no longer there, like blood vessels, for example. In this picture down here, we see an artery and a vein. The artery often will remain open when it's been cut, and that's because the wall of the artery has a lot of muscle to it, and the muscle lends strength to that wall, and it will hold the artery open even when there's no blood. The vein, however, veins do not have as much muscle in the wall, and it's mostly just the presence of blood in the vein that hold it open. So when you section a tissue with a vein in it, there's no longer any blood inside the vein, and so the vein collapses, and it looks like that strange sort of smile structure when it's been cut. Now there's four main tissue types that form all of the structures of the human body: epithelial tissue, connective tissue, muscle tissue, and neural tissue, also known as nervous tissue. And I like to talk about skin when we're talking about these four tissue types, because skin is an example of an organ where we can clearly see the four different types of tissue. So again, an organ by definition is a group of different tissues working together for a particular function. So in skin, we see epithelial tissue that's on the surface of skin called the epidermis. It's very protective. Deep to the epidermis, we have several layers of connective tissues that help to connect the epidermis to the underlying tissues. We also have muscle tissue in skin. Not a lot, but a little bit. There's a little bit of muscle that's attached to our hairs, and this is called the erector pili muscle, and it causes your hairs to stand on end, which we call goosebumps. And then there's quite a bit of nervous tissue in skin. Most of the nervous tissue is there for detecting touch, so things like pressure and light touch and that sort of thing. But some of the nervous tissue is also feeding information to the erector pili muscle, for example, or to various glands too. So just a quick overview of these 4 different types of tissues. So epithelial tissues tend to be found covering exposed surfaces of the body. They're very protective in nature for the most part. We also find epithelial tissues lining passageways where skin goes in, right? So like the mouth, vagina, urethra as examples. Epithelial tissue also will form glands, so epithelial tissues can secrete substances. Now, connective tissues are different. I think of connective tissues as like the junk drawer of tissues. If it's not an epithelial tissue or a muscle tissue or a nervous tissue, it kind of gets thrown into this junk drawer category called connective tissue. Now, a lot of connective tissues do connect, but some of them don't connect at all. So it's called the connective tissue group because most of them do connect. Connective tissues are the most abundant tissues in the body, and some of them do connect, like areolar tissue is the true glue of our body, but some of them don't really connect at all, like bone tissue or blood. Muscle tissue is specialized for contraction. And so this is the tissue that will be used to perform all of the movements of the body. So like cardiac muscle tissue in the heart for moving our blood, skeletal muscle tissue, which is attached to our bones for moving our body, and then there's smooth muscle tissue, which is found in the walls of our hollow organs. And this will help us to do things like propel food through the digestive tract, move urine, right? It's found in the walls of our blood vessels to help to regulate our blood pressure. And then last is neural tissue, also called nervous tissue. And this particular tissue is specialized for high-speed electrical and chemical communication. Very important for homeostasis of the human body.